Everyone, I've made some new friends today, so happy to see everyone here. This is going to be the introduction to gene therapy talk, and I'm delighted to have Eileen Sawyer here from Swan Bio as well. And so together we're going to take you through a brief overview of gene therapy. That'll probably take about half an hour, and then I want to open this up to questions and hear your questions. And again, this is a new field, so there are no wrong questions. Don't be shy. If something in your mind is unclear, then others will be likewise having questions. So I would love to make this a little interactive and chat with you about where we are, where we have been, where we are, and where we might be going. And it's really pertinent to you, so you are the ones that we want to hear from. We will be focusing mostly on adrenal leukodystrophy and highlighting two different approaches to gene therapy. And the goal of this session is a simple one, that you can distinguish between these two different types of gene therapy. And there'll be a quiz at the end. No, just kidding. So, here are my disclosures. So I know this is probably small print and hard for people to see, but just to highlight that the history of gene therapy has been a long time in the making, starting with the 1960s discoveries of Adeno-Associated viruses, cloning and sequencing of the AV2 genome, first applications of what's called a human retroviral cell-based therapy. And it was then in 2009 that the first human neurodegenerative disease was treated with a lentiviral vector. And that happened to be experiments by Patrick Aubourg, Nathalie Cattier, on whose shoulders we're standing today. But importantly, over the past few years, there's been a huge surge in efforts around AAV-based gene therapy and FDA drug approvals around AAV gene therapy for eye disease, as well as for spinal muscular atrophy, another disease of the spinal cord. And so while it seems like a lot of things are happening right now, they've been decades in the making. And just to give you that sense that there's been a lot of basic science discovery over the years that have led us to where we are today. So I will be using some words here that might be new and unfamiliar to you, but just keep note of those and ask me afterwards. So these are the two different types of gene therapy that I want you to understand afterwards. One we call ex vivo, which means outside of the body, and the other we call in vivo gene therapy, which is inside the body, okay? So ex vivo, outside the body, in vivo, inside the body. And the ex vivo that I listed first uses a certain kind of virus. It's called a lentivirus. And the in vivo, inside the body, uses an AAV-mediated gene therapy. So as I said before, there are different kinds of viruses and viral vectors. And the first thing we can say about these viral vectors, and bear with me, I know no one wants to hear about viruses these days, but these viruses are attenuated. That means they are being made safe, and they are different in size. There are adeno-associated viruses that are smaller and lentiviruses that are larger. Okay, so the adeno-associated virus you can see in green here is small, what we call a DNA parvovirus, whereas the larger lentivirus is an RNA virus. And they have different kinds of properties, okay? They may belong to the same family of viruses, but like in any family, they're the wild ones, they're the mild ones, right? But here it's around the nature and personality of these viruses. And the AV virus has a particular property of liking to go to the nervous system. And so it can go to different parts of the brain. There are certain different kinds of viruses within that family that have a predisposition to go to certain cell types. Lentiviruses, on the other hand, they tend to integrate into the genome, which means they deliver well into the bone marrow cells, into what we call stem cells, from which continuously new cells are being generated, okay? So don't get too confused about the terms. Just remember, there's in vivo gene therapy inside the body and ex vivo outside the body. And we usually use two different kinds of viruses, okay? Same thing again here. On the one hand, in vivo gene therapy, where we have a direct delivery of the AAV carrying the transgene to a certain organ, like the liver. We directly deliver that virus carrying the cargo, carrying now a healthy copy of the gene. On the other hand, cell-based delivery, usually you harvest cells from the patient you're treating, and then inside a dish outside of the body, you transfect, you use that virus to now deliver a healthy copy of the cell. And then you take the cells, this is not the naked virus like over here. This is the cells that are now going into the body, okay? So you can just, if you think of this picture, maybe it makes a little more sense. One is delivering the naked virus to the organ, and it carries a healthy copy of the gene, and that is now delivering directly. Whereas this is an indirect delivery because it's actually depends on you taking cells outside of the body, transfecting them and giving them back. Okay? Now, depending on that, you can imagine that there are different delivery routes and target cells, as I mentioned. So for anything that is delivering the virus directly, you might have to inject it into the brain. You can deliver it into the CSF space, similar to a lumbar puncture. And Eileen Sawyer will talk more about this. On the other hand, the ex vivo gene therapy approach is something where you are usually taking the cells from the bone marrow, transfecting them in a dish, and then delivering them back. Now, why am I talking about these two different approaches? Well, it turns out that even a single gene disorder, like adrenal leukodystrophy, has different manifestations. As we've talked several times in the last few days, you can have cerebral ALD that affects the brain, or you can have AMN that affects the spinal cord, right? So these are different parts of your nervous system that are affected. And depending on what part of your nervous system is affected, I'm going to argue you need a different kind of gene therapy approach, okay? Not one size fits all. Just to, again, remind people, childhood cerebral ALD, you can see the lesions in the white matter of the brain. And here are certain cell types that are very inflammatory. The blood-brain barrier opens. you can deliver cells from bone marrow to blood to brain. In AMN, that's not the case. We actually don't have an open blood-brain barrier. And here it's these long tracts that are going up and down in the spinal cord that we need to deliver to. And the simple answer is doing this through the CSF is much easier and delivers directly into the spinal cord without having to navigate its way all the way through the bloodstream. Okay, so I will be focusing here mostly on cerebral ALD in the first part, and then Eileen will come and speak about an AAV approach to spinal cord. So far, so good? Okay. So here now a little background on why we're doing ex vivo gene therapy. Many of you know that for decades, we've been using bone marrow transplantation for childhood cerebral ALD, right? And this happened less out of an understanding of how the disease works, but more out of let's try this for a bad condition, similar to transplants having been used for bad diseases like cancer. You know, you throw the kitchen sink at it and you see what works. And maybe we have a chance at modifying a devastating disorder by exchanging the bone marrow. And in many diseases it didn't work, but in adrenal leukodystrophy, it did work if you did it in the early disease stage. If you did it at a stage where there was a small lesion growing and you had a good bone marrow donor, which means either a matched sibling donor or a donor who was had a good HLA match, as we call it, meaning the cell types were well matched. Okay. However, there still remained several risks and limitations. You know, as you heard in the last few days from our transplant colleagues, there's still transplant-related mortality, graft failure, graft versus host disease. So the idea came about to say, instead of taking somebody else's cells, let's take the cells from the boys themselves. Similar to the picture I just showed you before, where you're taking cells from the bone marrow of the boys and now correcting them and giving those cells back to the boys themselves. So this is exactly the kind of trial that we developed. There was a small lesion in the brain that showed contrast enhancement. We collected hematopoietic stem cells, meaning bone marrow cells from the boys. We used one of these lentiviral vectors, transfected the cells with delivering a healthy copy of the ABCD1 gene. Okay, right? We're talking today about that address in your ABCD1 that was causing it not to work well. Well, you can actually deliver into cells a healthy copy where the address is intact. And then you deliver those cells back to the boys after a little bit of chemotherapy using busulfan and cyclophosphamide, or busulfan and fludarabine. And the chemotherapy essentially makes room in the bone marrow, okay, so that the new cells can come in. And then you watch the boys. And the idea is that these new cells in the bone marrow go from bone marrow to blood and from blood to brain. And this whole procedure is actually very similar to what happens with a conventional bone marrow transplantation, with the only difference that you're now using cells from the boys themselves. So what are you avoiding? You're avoiding engraftment problems, you're avoiding graft versus host, all those reactions to foreign cells. You don't see that anymore, okay? So that's the advantage here. And I'm just going to summarize briefly here that this was, for the most part, well tolerated. Most of the boys did very well. There was one boy who was already progressing very rapidly at the time of treatment, and he sadly succumbed to disease progression. This is a neurologic function score where the higher the numbers here, the worse you are, and the more symptoms you have. But you can see that most of the boys did very well, and we've treated actually up to date 67 patients worldwide. So it's one of the largest gene therapy trials. So what happened to the MRI lesions? Again, this is a scale where the higher numbers show higher lesion load. And you can see that in the first year, the lesions still grow, but by the second year, most of the lesions start to stabilize over time. And that contrast enhancement that often was a trigger for treatment, that also got better and improved over time. So what we did notice, and I know this is a busy slide, so I don't want you to look at all these complicated words, but I want to point out that there were 3 boys who had something we call Myelodysplastic Syndrome. Now, what is that? Myelodysplastic Syndrome is a long, complicated word for saying that your bone marrow doesn't produce blood cells the right way. And so we noticed this because the boys who came out of the trial, those 3 boys, they tended to have low blood counts. And that made us suspicious and made us then analyze the bone marrow more carefully. And these boys have a bone marrow that now doesn't make in particular platelets and other blood cells well. And they've required now a second transplant. But what I want to point out is that all of these boys have remained neurologically stable, okay? It's actually not affected their neurologic function to date, and they still have a 0 on their neurologic function score, which means they don't have any symptoms. So that has been a big relief for me as a neurologist. But it is definitely something that teaches us, while we made a lot of progress in delivering a healthy copy of the gene back, and we were able to stabilize the disease in the large majority of patients, there are still risks here. And we have to be aware that we might have traded in, a very bad neurologic disease for some changes now in your blood that may look a little bit like cancer. And so we have to be very careful. So as of January 31st of this year, we've identified 3 cases of the total 67. So that's about 4%. So still a low number, but we say we have to be careful. We don't know, this number might grow. And we think it's because of this particular lentivirus that the myelodysplastic syndrome occurred. There's one more patient who has not got a low blood count, but has a clone, and we're following him carefully. That means that there's a certain population of his bone marrow cells that has started to expand. So a lot of this language is a little technical, but I just want you to take away from this that there are 3 patients who had this blood disorder and had to undergo a separate transplant. But they've no indication now of this blood disorder on last bone marrow assessment, but we're keeping a close eye on them. We don't know if they're out of the woods yet. So that having been said, most patients with cerebral ALD have benefited from this gene therapy, and we have to carefully weigh the risks and benefits here, okay? But a quick update on the ex vivo lentiviral gene therapy for cerebral ALD is that 2 weeks ago, we were delighted to hear that the FDA advisory committee unanimously endorsed this new therapy for early active cerebral ALD, meaning that they recognize that while there are some risks, the benefits currently still outweigh those risks, okay? That there is still more good that these boys can experience by getting the corrected cells than the potential risks. So I'm now going to turn over to Eileen, who's going to now move from ex vivo gene therapy outside of the body to in vivo gene therapy, okay? And then we'll have questions after.
All right. Good afternoon, everyone. My name is Eileen Sawyer, and as Florian said, I'll be talking to you about specifically the in vivo gene therapy method using AAV. And it's really a big pleasure to be here to talk to you about this, and especially to be sharing the stage with Florian. So I work for a company that's working on developing this type of gene therapy for neurological disorders. It's a small gene therapy company that was founded back in 2018 by a trio of experts that each had a specific expertise. One is a neurologist who really knows AMN and ALD very, very well. And they came together with a scientist who knows AAV gene therapy very well, had been working in the area for over 25 years, together with a patient expert who knows the disease of ALD and AMN very well. So what I want you to understand from this is that in order to develop a gene therapy, it takes a lot of different types of expertise to come together in order to actually bring one of these therapies out of the lab into the clinic and hopefully at some day into medical practice as well. So Swan Bio is focused on specifically this type of in vivo AAV gene therapy with a lead program called SBT-101, which I'll tell you a little bit about today, in development for the treatment of AMN, the adult form of ALD. And we're hoping by targeting the spinal cord in the situation of AMN that we'll learn about how to do this well and be able to then take that knowledge and apply it to other disorders that might benefit from this same type of approach. So Florian did a very nice job taking you through these two different types of gene therapy, the ex vivo, which is on the gray side of the slide here, and the in vivo, or direct delivery, which is on the purple side. As he described, the ex vivo, or cell-based delivery, involves taking cells from the patient, modifying them, and then putting them back into the patient, where the direct delivery form, you can deliver the gene therapy itself directly into the patient's body. And so that's what I'm going to focus on, and I'm going to go in a little more depth as to exactly how that works or how we believe it works. Florian had also mentioned that the AAV class of medicine has actually been around and being studied in humans for over 20 years. So it may be new to this space, but there have been thousands of patients across many, many different types of disorders that have received a version of this type of medicine. And this has really taken off in the last 5 to 7 years as more and more of these therapies have made it into the clinic and being studied in people. And in fact, 2 were approved by the FDA. There was Zolgensma, which you may have heard of back in 2019, which is an AAV9-based gene therapy for children with spinal muscular atrophy. There's also Luxturna, which was the first one approved in the US back in 2017. That's an AAV2 gene therapy that's for a form of inherited blindness. And there are many others that are coming down the pipeline towards late stage development as well. So over these last decades of studying this type of medicine, we've also learned some things about the overall general safety profile, and what you might expect when you give a patient one of these types of gene therapies. And there are two potential areas of reactions. The first is in the immediate period after you give the gene therapy. The first 1 to 30 or so days, there's the potential for the immune system in the body to have a reaction to the vector because it's made with some pieces of a virus. So your body might mistake it for a foreign invader and mount a reaction the same way it would if you were infected. So sometimes, depending on the type of administration, whether it's administered to the whole body or directly to a certain part of the body, what the dose is, you might see things like a reaction in the liver or some general tissue inflammation. And at this point, these reactions are fairly well described, and many of them respond well to management either with steroids or another form of immunosuppression. In addition, as these therapies are fairly new, there's also the potential as we treat more patients and follow them for longer periods of time, we'll uncover longer-term reactions. So far, these have not really been observed in the patients who have been treated, in some cases for up to 5 or more years. But there is a risk, even with these types of therapies, that you could get an accidental integration, as it's called, into the patient's gene, even though that's not an intended effect of this type of viral vector that could lead to cancer. So this is something that patients are monitored for over time, but like I said, it's yet to have actually been observed in patients. It's only been seen in certain types of animal models to date. So these two categories of safety events are something that are understood to be a potential effect when you're treating with this type of medicine. And so patients who receive it are monitored for both of these types of reactions. So let's take a closer look at exactly what is an AAV vector. Adeno-associated viruses, the viruses themselves, are naturally occurring very small viruses that on their own don't cause disease in humans. So to make one into a vector, we actually take the different components of it, and we use the outer protein shell, which is called the capsid, as a delivery vehicle for the genetic cargo, or the gene cassette. And this is what allows us to directly administer it into the patient and for it to find where it needs to go in the body, whether that's the cells in the CNS, the cells in the liver, or the cells in the muscles, somewhere else. So there are many different types of capsids. They're called serotypes, and depending on which target you have, you may choose a different serotype. You can think of it like the cardboard box that a package comes in. That outer shell, that protein shell, it's got the address of where it needs to go in the body and gets delivered there. And then once it arrives at the cell, that's what this slide here shows, is the blue circle is meant to be a cell in the body. So when it arrives at the right cell, the cell takes it in just the way you'd take that package into your house. It takes off the wrapping, takes off that protein shell, wads it up and throws it away. It doesn't need it anymore. And what remains there in the nucleus or the center of the cell is that gene cassette. And the gene cassette contains the instructions for what we call the therapeutic gene, or the gene that we believe would be helpful or beneficial for your body to produce to help ameliorate the effects of the disease. And so that stays in the nucleus, and it comes with the instructions on when and how to read the gene to produce the molecule that we believe to be helpful. And it can be used again and again and again by that cell. So it does not generally integrate into the DNA that you're born with. It remains in a little circle called an episome sitting outside that. And it's stable there as long as that cell is alive, as far as we understand. It will not get passed down to your children. It stays in your cells. It does not get passed on to any children if you have them afterwards. So SPT101, which we're developing for adrenomyeloneuropathy, is a version of one of these AAV gene therapies. It's using an AAV9 type capsid, which is known to be effective at transducing the tissue in the nervous system. And it contains a working copy of the ABCD1 gene, which is the one, as Florian explained, is impacted in ALD and AMN. And we're intending to administer this intrathecally, which means into the cerebral spinal fluid that is in your spine and bathes those long tracts that are affected in AMN. And we chose AAV to target AMN for a couple of reasons, as opposed to the ex vivo type that Florian was describing. As I mentioned, the AAVs can target and deliver that genetic cargo to the types of cells that are in your spine, and those are the ones that are affected. And because the cells in the spine, well, they're difficult to access, we couldn't take them out and transduce them and put them back in. We have to access them where they are in your spine. But they also live a long time. So if we can get them the healthy copy of the gene, they'll probably be able to retain it for a while and use it. So this is part of why we chose the AAV as the approach to the spine. And last but not least, it's easy to access the spine directly. So we're pretty confident that we can get the vector to the cells that need it. And that's through the intrathecal injection. So this is like a lumbar puncture. The doctor would insert a small needle into the spine where they can infuse something directly into the fluid that's around your spine. And this is a very common type of procedure to do a lumbar puncture. For example, an epidural that happens during childbirth uses a very similar approach of injecting something into the spinal area. And so this is how this would be delivered, and it also helps ensure that that drug goes directly to the areas that need it before it goes anywhere else in the body. So to wrap things up, a couple of key things to remember about AAV gene therapy. They are generally administered one time and deliver their gene to the cells where hopefully that will stay. And this is that in vivo gene therapy delivered directly to the patient. The genetic cargo does not integrate or modify the genes that the patient was born with, again, unlike some of the ex vivo gene therapy, and it's not passed on to a person's children. In fact, neither of these types are. And we are planning to initiate studies with the AAV-based gene therapy for AMN later this year. So I don't have any data that I can share today, but hopefully in the years to come, we'll be able to. All right, and so thank you. I think now we were going to open it for questions.
So does anyone have questions?
Thank you. That was great. The blood disorder that happened in the couple of boys, you said then you give them another transplant and it won't come back. So it's like something that happened initially. It's not something they have that they're going to get again.
The shorter answer is we don't know long-term what will happen, okay? But it looks like the transplant that they received corrected the problem. But it is still early days. There's a lot of uncertainty about this. We also don't understand what the exact nature of this blood disorder is and whether it acts in the same way as other myelodysplastic syndromes, where it's a precursor to a more malignant cancer or not. So there's, still small numbers and early days, but there's a lot of research happening on this, and I'm sure in the coming years there'll be more insights.
And has it happened in other ex vivo gene therapies?
Good question. So this particular type of myelodysplastic syndrome has not occurred in another program that's happening currently for thalassemia. It's not been seen, but there have been some different kinds of events happening in another sickle cell program. So we do think it has very much to do first with the lentiviral vector and probably also with the type of promoter that's being used. We think that promoter is what made this treatment so effective. And so that was one of the reasons to stick with it. But time will tell whether we need to change that or whether it's going to be hold its own.
We have a Zoom chat question. Is there a difference between the two types of therapies discussed today in CRI-SPR? If so, how is CRI-SPR based therapy administered?
Thank you. So CRISPR is a type of system that is used to edit the genes that a person was born with. And it can actually be used in either an ex vivo setting, and there are even studies now using it in vivo settings. Those are starting as well. So the difference is that CRISPR is the system that's used to affect the change, and the ex vivo and in vivo systems that we're talking about are just delivery vehicles.
So I'm curious about what the aftercare or follow-up care looks like in gene therapy, and if it differs, whether it's in vivo or ex vivo.
So I'll start and then let Florian comment. So the aftercare is going to look different for every single gene therapy. They'll be tailored to the type of disorder it is, exactly the type of gene that's being delivered, and then the system as well, whether it's ex vivo or in vivo, and even maybe some additional nuances there. There are some generalities that you can take. One is that, as I mentioned, it's very common for immunosuppression or steroids to be used after an in vivo AAV gene therapy, but exactly what that looks like, how long, what type of immunosuppression, that's very dependent on the program and the patient.
Yeah, so what else to add here? What FDA requires from us is for some forms of gene therapy, like the in vivo gene therapy, there's a 5-year follow-up period that requires close monitoring. It's even longer for the ex vivo lentiviral gene therapy, which is 15 years. Some of this has to do with monitoring for the kinds of events that we saw many years out. And understanding what they mean for patients and how to manage and handle it. As Eileen said, in the in vivo gene therapy, it's more around understanding, the need for immune modulation over time and whether steroids would have to be reintroduced or not. But I'm thinking overall, as we're making progress in gene therapy, it's becoming a field where we are learning the tools to manage this step of gene correction, which is, of course, a huge undertaking in of itself, and not surprising. We have to monitor and understand when to use tools to modify things. Did that get to your question, Lisa?
So when you talk about the immune and inflammatory reactions to the in vivo gene therapy, is that similar to GvHD with bone marrow transplant, or is that different?
It's different.
Okay.
So inflammatory reactions that we see with in vivo gene therapy, I must say, also differ depending on where you're delivering to, what compartment you're delivering to. There's less immune response is seen if you're in an immune privileged compartment like the eye or the spinal fluid. But if you're delivering into the blood and to the systemic circulation, the immune system is right there and sees and reacts to these viruses. And honestly, we're treating children, little babies here every month now for some devastating leukodystrophies that we previously would never have been able to treat. And we're seeing some immune responses, which is telling us this AAV is doing something, and we need to give steroids and watch the liver and inflammation, and as it rises, understand, when will it stop and when will it go back? Do steroids need to be increased or not? This is very different than graft versus host or engraftment problems that are a reaction to a foreign cell coming to the body. And this is something that we do as an inpatient with a transplant team, a different set of immune modulatory drugs as well.
But you could use steroids for both of those, right? Because graft versus host, I think you can use steroids as well.
Yes.
Because I know a lot of the boys are on that for many years.
Yeah. The steroids are used for so many different indications, but in general, sort of graft versus host disease requires different kinds of is of a different nature and use different kinds of immune suppressants rather than steroids. But you're right. In both cases, we're also dealing with probably consequences of how long can we use steroids and when can we start tapering them, because they do have some long-term effects.
We have a few more from Zoom. Is there any biological marker except for clinical and radiologic markers in blood or CSF looking for to make sure that gene therapy doing well in AMN with AAV9 therapy? Repeat it. Okay. Is there any biologic marker except for clinical and radiologic markers in blood or CSF looking for to make sure that gene therapy doing well in AMN with AAV9 therapy?
So I'll do my best to answer the question and then maybe ask Florian to comment as well. So I believe the question is asking whether there are any markers in the blood that we can look at to see if an AAV9 gene therapy, in vivo gene therapy for AMN is working. So that's a very good question. We are in the process of starting up the first trial in humans. So I would say we're going to be looking at a lot of different markers, and we'll have a much better idea of the answer to that question in the future when we have a little more experience. Florian, I don't know if you have anything to add?
Yeah. So across neurological diseases, there's a lot of biomarker discovery happening right now, and a lot of excitement around markers such as neurofilament light chain and things that might be hopefully an early indicator of us, changing the course of AMN. But it is a challenge. It's we're dealing with a disease that's not in the systemic circulation, and we don't have an open blood-brain barrier here. So we'll probably be mostly relying on sampling spinal fluid and and looking at imaging to make sure there's everything safe. But ultimately, it's going to be clinical outcome that's going to be critical.
Any other questions?
We do have more from Zoom. We have about 5 minutes remaining.
I know some of this may be speculation at this point as we're just getting started with AMN, but what are your thoughts or what are our knowledge right now as far as the success of these treatments in various stages of disease progression?
So that's a great question. And, it's one that I hope that over the years we'll be able to answer more fully than we can today. When starting out with a gene therapy like the one that we're talking about, initially we want to investigate it in a population where we're going to be able to tell the safety and see if it has any effect on disease as quickly as possible. So in that case, we're often looking early on in the disease with the hope of being able to show some type of slowing or halting of that progression, because the mechanism for that is probably a little bit different than one that would, say, be reversing injury that's already been accumulated in the spine. So it's a stepwise process of trying to understand the effects over time, and then be able to say, okay, it's able to do this or not that, and explore those different avenues.
Yeah, no, that's right. And I'd just add to that the fact that we don't see frank cell death in AMN is very encouraging. There's not been many autopsies, but some of you might have seen Jim Powers here last night. I mean, he's the one who first described spinal cord disease in AMN, and the cell types that are next to or in the spinal cord are alive. They're under duress, but they're alive, and there's a real good potential that if you correct the gene, that those cells will recover and be fully functional. And in the mouse model, we also haven't seen any sign that the AMN mouse has cells that are dead. So as long as the cells are viable, we think there is a good chance that we can deliver this cargo safely and maybe even reverse things. But it's early days, so it's hard to know whether that's possible.
Any other questions?
We have about 2 minutes. We have one more. Do you think something like this would be able to help CTX patients?
Good question. CTX is also a single gene disorder, and it's also a recessive disorder where gene addition might be helpful. So, there's no reason to think that this may not be possible. Again, any therapy for a disease has a population where it works best and a population where it might be contraindicated. So you want to be careful and choose that stage of the disease wisely, because not one size fits all.
Is that it? One last chance. Questions? All right. Thank you very much.
Is this where the quiz starts, Chris? No.
Oh, yeah, absolutely. I'll pass them out now.
It's good. Thanks, everyone, for attending.